EasyGDB includes multiple bioinformatics tools, such as the search by gene ID or annotation keywords, BLAST, genome browser, sequence extraction, annotation extraction, or gene version lookup. The search tool is always available in the toolbar. The results can be exported in several formats. The columns of the table can be filtered and sorted. And new searches can be done to filter the results. The genes are linked to the gene annotation page. Here we can see the gene in the genome browser. Find all the annotations associated with links to their sources and the sequences of the genes, which are taken from the BLAST database files. In BLAST, we can find homologous sequences based on sequence similarity search. The input type, nucleotides or amino acids, is automatically detected. BLAST databases are also automatically detected, and just adding the files in the BLAST-DBs folder will display new datasets in the BLAST tool. Results can be downloaded in tabular format. Graphs display query coverage, score, identity percent, and descriptions. And genes are linked to the gene annotation page. Alignments and more metrics are shown below. JBrowse is used as genome browser, which allows multiple species implementations and track visualizations for many data types. Here, genes are also linked to their annotations. Other tool is the sequence extraction tool, where you can paste a list of genes to get their sequences. Again, the sequence datasets are based on the BLAST files in the BLAST-DBs folder. We can also extract annotations for a list of genes using the annotation extraction tool. This is very useful to add annotations to the results of your analysis. Annotations can be exported in many formats, and for example, can be easily pasted in an Excel sheet keeping the order of the genes. In this table, columns can be filtered and sorted, and in the search box, keywords can be used to find annotations. Again, genes are linked to their annotation page. The last tool is the gene version lookup tool, where you can find other gene versions or homologous genes from other species based on a list of gene relationships. Genes with a relationship of 1 to more are displayed in a pinkish background.